So today we will continue our lectures about molecular biology, and we will be talking about epigenetic mechanism of gene expression. And as I told you in my first lecture, that there's two forms of chromosome. It can be loose or it can be condensed, and if it's loose, it's actually active and it's able to transcribe its genes and make proteins by translation further. So there's actually epigenetic mechanism, which means it does not damage or change DNA structure, gene structure itself. It works out of gene. So we usually use chemical reactions such as methylation, acetylation, or deacetylation to change DNA form to make it more condensed or more loose and be able to be expressed. So here we have histone octamer, which contains of four histones, which is four types of histones, but eight of them, like each type is duplicated, and it makes like octamer structure, which will loop DNA twice and make this nucleosome first, right? And it actually happens when it's methylated or deacetylated, and it's reversible process. And by doing that, we making gene active. Like here, you can see that it is not bind with histone and actually able to be expressed. And here, it's not active because it's bind to histone structure. So how do they bind DNA? The DNA and histone have different charges. Like DNA charged negatively because it contains phosphor, and histones are charged positively because it contains positively charged amino acids such as lysine and arginine. So because of that, they will actually combine and work together. Beside histones, UA will actually methylate DNA itself by methylating a specific site like promoter gene, which is CPG, cytosine guanine. P refers to phosphate gene side of the gene. So when you actually methylate this, you make uh, stop you stopping the excitation, expression of the gene by silencing it. You silencing gene expression. So here we have a page from first aid which shows you nucleus. It's not a cell, entire cell, it's just its nucleus. So here we can see like condensed form of chromosome, part of condensed part of the chromosome, which called heterochromatin, and we can see loose part of the chromosome, which called euchromatin, and which site is active, where transcription occurs here in euchromatin, because when it loose, it it actually able to be translated to express itself, right? And here we can see nucleus, which is on the center. And when we talk about nucleus, con- actually contains rRNA because it usually synthesizes RNA for ribosome, and it actually makes ribosome structure. But translation occurs only in cytoplasm, not in the nucleus. Even ribosomal proteins go like first they will be synthesized in the nucleus, not nucleoli, right? In the nucleoli, we have only rRNA synthesized. So then it will go out, translate there, and we already now have mature ribosomal proteins, which will come back to the nucleus, go to the nucleoli, combine with rRNA, and make mature um, ribosome. Part of mature ribosome will go out, part will stay in the nucleoli. And heterochromatin is usually located on the periphery, and we actually have specific type of heterochromatin in women. It calls Barr body, and it forms when one X chromosome is deactivated. Here you have more darker structure, which is super condensed, not able to be loosed again. It is Barr body. It's like entire X chromosome methylated and deactivated. Here you can see like this one is heterochromatin. It's not as condensed as this one, and it's not as dark as this one. And here in males you can't see any Barr body. There's also a disease associated with pathologic methylation of DNA. It's called fragile X chromosome. It's called fragile because of the side, like methylated side make DNA look like it's fragile, and this part can fall off anytime because these parts are condensed. As you can see, in patient will be present with large ears, with big forehead, with mental retardation, with and microorchidism. And when we talk about histone methylation, it's actually methylates specifically lysine and arginine residues. And again, histone acetylation will remove histone positive charge, and it will release DNA coiling, and it will be transcribed then. Uh, on the other hand, when you deacetylate histones, it will remove acetyl group, and it will actually tighten DNA coiling and decrease transcription. And there's actually disease associated with altered gene expression because of defective deacetylation of particular parts of the chromosome, and it's called Huntington disease. Here you can see pathological sites which cannot express, and because of that, pathologically mutated Huntington protein will form, and it will cause actually in early stages of the disease, like you can mistaken this disease with alcoholism because patients are unable to, um, they can swallow, their hands are shaking, and they have kind of a toxic gait, and later on the disease stage they can develop Parkinsonism. Here again you have like entire cell, you have nucleus, and within nucleus you have euchromatin, you have heterochromatin, you have nucleoli, which is composed of ribosomal proteins and rRNA. And ribosomal proteins are synthesized once again in the cytoplasm, and then they will go back to the nucleolus to form like mature ribosome. So here we can see structure of one nucleosome. It contains like four types of histones which are duplicated, and you have eight histones which will loop DNA twice around itself, and then you will fix all of that with histone 1, and actually histone 1 will attach to specific like side of the DNA. It's called linker DNA. So what the point of epigenetic mechanism? It's actually makes variation between cells and between tissues and because of different structure, and it actually makes unique functions for a specific type of cell or specific type of tissue, and it's actually responsible for imprinting, which is when one parent can give only certain genes expression and other genes will be um, silent, and therefore they have to take silent gene in, like, the part of silent gene in one parent from other, and it actually will make like the whole new organism, functional organism, and therefore you'll enable to make a viable organism from two sperms cells in the tube or from two ovum oocytes in the tube. So it's actually also responsible for cancer development because it regulates like expression of anti-apoptotic and pro-proliferative genes, and it silences tumor suppressor genes. And now we'll talk about genomic imprinting and its inactivation. So what's are similar here? Both are caused by inactivation that stay in place for the life of the individual. And what the difference is, X inactivation turns off entire chromosome, whereas imprinting turns off only specific genes on one chromosome, right? So genomic imprinting is the process by which only one copy of a gene in an individual, either from their mother or their father, is expressed while the other copy is suppressed, as I said. So instead, gene expression is silenced by the epigenetic addition of chemical tags, methyl groups, to the DNA during egg or sperm formation. So epigenetic tags on imprinted genes usually stay in place for the life of the individual as well. So X inactivation, on the other hand, it's called also lionization because of British genetic Mary Lyon, which first described this process, right? So it's X inactivation just ensures that females like males have like equal amount of chromosomes because male have only 45 X chromosomes, whereas females have 46 X chromosomes. And in order to survive, females as well have to have 45. So what we do, we just inactivate one chromosome. And by inactivating it, we actually form this Barr body I talked before. And we actually have diseases which contains more than one X. When it's male and it's filter, Klinefelter. It actually has Barr body and it's normal male, but it's actually mutilated and formed Barr body in male. It's like the only example where you can see Barr body in male. So, and if you have triple X, you actually have two Barr bodies and one active X.